All right, welcome to our second GM of the quarter. Let's jump into announcements. First, as you know, our attendance policy, you get 2 excused and 1 unexcused absence per quarter. And this covers GMs and committee meetings. So please let your beautiful directors know 24 hours in advance. Number 2, you should have received this in your inboxes, but please fill out the membership renewal form so we can keep track of all of our active members. And we'll be removing inactive members from the Slack channel. So please fill it out with your name and email. And lastly, we have our mentorship application. So apply to be a family head with a fellow hand mentor we'll pair you up with. And you two will be co-leading a family of mentees. So please fill it out by Friday midnight and we'll start sorting out the families. Okay, I'm gonna pass it to Sasha for our icebreaker activity.
Yeah, hey everyone. So we have a fun icebreaker plan for today. And this is just an activity that we come up with and it's entirely experimental. So I'm interested to see what you guys come up with. So Tiffany, if you want to do the next slide. Yeah, so this is a game called Emoji Story. And next slide, please. Yeah, so what we're going to be doing is everyone's going to be separated into 6 different breakout rooms, and you're given a set of 10 emojis each. The goal is to come up with the craziest, like funniest story that you can possibly make that ties all of the emojis together. And the goal is basically to flex your sense of humor. The only rule that there is every story has to end with a line, and that's what I did on New Year's Eve. So I'm really interested to see what you guys come up with, and I'm gonna put a link in the chat real quick. This is a Padlet, so you guys can just put your stories on there. And we'll basically be doing like an informal voting on who came up with the better story for their set, because there's 2 people, like 2 rooms that are gonna be assigned to each set. Cool, does anyone have any questions? I don't think it's particularly complicated. We're gonna see how it works out. I'm like very interested. And as RT said, I really, really encourage you all to turn your cameras on and just kind of have fun with it. Cool.
Great, you send the Padlet link now. Okay, great. Also, if you haven't already, please screenshot the slide so you can reference in your rooms. Okay, we're gonna have 6 rooms, teams of 4 to 5 people. Okay, have fun, get creative.
I have a question. Should I be joining a breakout room or not?
Yeah, go for it, Sasha.
Should I go for it? Okay.
Just as just as an overview, Dr. Baton will be coming to our GM next week, and he is a professor at UCLA, has been for a while, I believe, and he does work in basically understanding the protein structures that cause Alzheimer's and Parkinson's and a lot of other diseases. So we're just going to be giving an overview as well as going over one of his papers through this presentation. So next slide. Yep, so our first topic is amyloidosis.
So amyloidosis is a range of diseases characterized by a buildup of abnormal amyloid deposits in the body. And basically what amyloid is that it is a protein that is abnormally produced in the body that can be toxic to various organs and body tissues, including your brain. The amyloid protein is insoluble and it can accumulate in the brain and disrupt its normal functioning. The picture at the bottom here, at the bottom right corner here is a PET scan that shows amyloid plaque building up in the brain. And the rightmost brain scan shows a healthy brain, whereas there's little to no buildup of amyloid. And the leftmost brain scan shows the brain of an Alzheimer's patient. And you can see a lot of amyloid building up that is shown in red. And next slide, please. So more on amyloid. This is a picture of the molecular model of amyloid protein fibrils. The amyloid protein is formed when misfolded proteins self-assemble into beta sheet structure. And this structure obtained by the amyloid is extremely strong, and they're highly ordered and organized, which makes the entire amyloid protein very stable. And in addition, the amyloid protein is insoluble, which means that it is resistant to degradation. And these factors combined allow amyloid protein to easily accumulate in the brain.
Yep. So one disease that is caused by or is identified by amyloidosis is Alzheimer's, and this is the most frequent type of amyloidosis. Amyloid deposits occur in the form of plaques and angiopathy. And so, like Z said, they're just basically build up in the brain that is not cleaned out. And then basically leads to cognitive deficits and degradation in the brain, ultimately causing memory loss and dementia. So if you look at the pictures on the right side, that's mainly what they look like. And so there's normal, and then if they break off, that's when they can start to become plaques and connect with one another and stop normal functioning in the brain. Next slide.
So age-related macular degeneration is basically a retinal neurodegenerative disorder, and it's characterized by what are called drusen deposits. So individuals with the disorder will experience vision loss, and it occurs due to these deposits that target the macula, which is basically a part of the retina at the very back, and it's responsible for nearly all of our central vision and color vision. So these drusen deposits have a lot of accumulated amyloid beta peptides as well.
Yeah, so to continue our overview, additional related diseases are myeloma, which I believe is a type of cancer, lymphoma, Whipple disease, Crohn's disease, and tuberculosis. And these are all, they're not necessarily caused by, but they do have a buildup of amyloid as part of one of its as part of its biology. Next slide. And so now we're going to talk about molecular tweezers and what they are.
So another really big concept that Dr. Bitten's research focuses on is molecular tweezers. And if you were in hand last year, you probably heard a lot about molecular tweezers in the research that we talked about and how they're being used to treat Parkinson's. So what molecular tweezers are they're artificial receptor molecules that are supramolecular, meaning that they're made up of two or more chemical species. So there's a picture of them on the right, and they basically have the ability to prevent abnormal protein aggregation, which is what causes diseases like Parkinson's and Alzheimer's. And if you look at the picture, you can see that there's kind of like two arms sticking out of the molecular tweezers at the end. And what they do is they bind the guest molecules, so like proteins, and because of that, the molecular tweezers are only connected at one end. So that gives them a sort of certain flexibility. And that's one of the properties that allows it to prevent abnormal protein aggregation once they bind to the proteins. And once they bind to the guest molecule, they're connected through hydrogen bonding, hydrophobic forces, van der Waals forces, and it basically inhibits the formation of toxic polymers in the body and aggregation of different types of proteins. And it just prevents these proteins from exhibiting their toxic effect on the body and preventing diseases like Parkinson's and Alzheimer's.
Studies using molecular tweezers have been done surrounding several diseases, such as Alzheimer's. In the early stages of Alzheimer's disease, a significant loss of synapses occurs, which results in neurons that are alive but are basically unable to communicate. This takes the form of a decrease in dendritic spines and electrophysiologic activity. Research in Dr. Bitten's lab showed how the usage of COR01 could be used to prevent these effects. In collaboration with the Frosti group at UCLA, they found that low dosages of COR01 reduce amyloid plaques and neurofibrillary tangles in the brain. both of which are considered hallmark lesions of Alzheimer's disease and interfere with nerve cell messaging. CLR01 was also found to be effective in suppressing the formation of AB oligomers and inhibiting the aggregation of tau proteins, which are a type of protein that are known to form toxic neurofibrillary tangles in the brain. Next slide, please.
So the development and research on molecular tweezers also impacts studies on Parkinson's disease, which is, as many of you know at this point, a movement disorder that causes tremors and like slowed movement and other cognitive deficits. So studies in Dr. Batan's lab have been conducted on alpha-synuclein, which is a protein that generally inhibits the release of neurotransmitters and causes a lot of issues for these patients. So in Parkinson's disease, these alpha-synuclein proteins aggregate into what are called Lewy bodies, which are abnormal clumps of protein in the brain, and it can kill the dopaminergic cells. So what was found through a collaboration with Dr. Bronstein's lab at UCLA, they found that this molecular tweezer called CLR01 was able to interact with alpha-synuclein and prevent accumulation in the brain. And basically what it does is it impacts the conformation of alpha-synuclein, so it's unable to like clump together, and it prevents that what is it called? The accumulation of the striatum specifically, which is the neuronal circuit that's responsible for voluntary muscle movement control. Sorry, not muscle control. Okay, so this next section is on biomarkers. So I'll just be doing a small intro on that. So the diagnosis of like CNS diseases rely on clinical evaluations, which have like a lot of variability like from patient to patient. And so this can lead to incorrect diagnoses. Most or a significant amount of diseases have similar deficits very early on. So it can be unreliable in distinguishing between those different things. Currently, there are two main methods that are used. So there's brain imaging and the analysis of cerebrospinal fluid. These are used for diagnosis, but they can also be highly inaccurate and they cost a lot. So MRI and PET scans are typically preferable. However, they're not accessible to all populations. And again, they're super high cost. So a promising approach that's been found is analyzing biomarkers, which are biological molecules that indicate normality or abnormality. And this is really what Dr. Batan's lab focuses a lot on. And these can be isolated from exosomes, which are basically like these vesicles that are found in extracellular space. And it can be isolated through like blood tests in like plasma and serum. Next slide.
Yeah, so now I'm just going to talk a little bit about how Dr. Batan specifically uses these exosomes and biomarkers in their lab. And so one way is that they're actually able to distinguish between Parkinson's and multiple system atrophy, which since they do have like certain similarities in clinical presentation, it can be hard to distinguish between the two. And so by measuring the levels of alpha-synuclein, they're actually able to account for these differences and distinguishing between these two diseases. And so they created this composite biomarker that included different variables, including the ratio of the alpha-synuclein in neural exosomes, and comparing that to an oligodendroglial exosomes. So the neuron versus the oligodendrocyte, which is a type of support glial cell in the brain. And they were able to actually yield an 89.8% sensitivity and an 86% specificity for separating the two disorders, which is pretty high, especially considering the difficulties that was discussed earlier with other methods. And it's also really great because it's just something that you can get from a blood test and you're able to find these significant differences. And then lastly, it's not shown on the slide, but they're also able to use these to measure the effectiveness of their techniques in terms of reducing pathology.
And so now we're going to be going into our short journal club. So if you had the opportunity to read the paper, it's called A Novel Molecular Tweezer Inhibitor of Alpha-Synuclein Neurotoxicity in Vitro and in Vivo. And it will basically concern that CLR01 molecular tweezer that we talked about. So we're going to be breaking you all up into 4 major groups, which are on the next slide. Yeah, so we'll have an abstract group, introduction, and 2 groups for the discussion. We're going to give you some time just to go over everything, discuss. If you need to quickly read over it, do so, talk about the major points, and then we'll all come together and we'll talk about just what were the major parts of the paper that you thought were interesting.
I'm creating the rooms right now. Okay, so we're gonna have 2 discussion groups, right?
Yes.
All right, wonderful. They should all be open right now.
Did not die. They survived. The embryo, because the alpha-synuclein aggregation was reduced and apoptosis was prevented. So they were able to translate those results and kind of conclude the alpha-synuclein expression was limited, or alpha-synuclein aggregation was limited with the use of the molecular tweezer. And this can be used to help treat neurodegenerative diseases such as Parkinson's, dementia with Lewy bodies, etc.
Awesome. Thank you, Sai. Anyone from the abstract group have anything to add? I think that was a really great overview of what was in there. Okay, looks like we're somebody in the chat. Yeah. So yeah, now we're going to go into the introduction. Anyone want to just shout out again what they thought was important about the intro?
I mean, real quickly, it's sort of like a repeat of what happened in the abstract, like in a bit of background, like on the protein, the alpha-syn, that, it's been implicated in all of these diseases, and that they have developed this CLR01 that is of interest. And they sort of go in at the bottom of like why it's of interest and what they're going to delve into for the rest of the paper.
Awesome. Thank you, Alex. Anyone else want to add anything that they thought was important? Okay, perfect. Yep. And just like the last 4 minutes of the meeting, we'll just go over the discussion. So one person from discussion group 1, and then one person from discussion group 2.
I'm not sure if we were group 1 or 2, but in our group we talked, or mostly what everyone else is saying, but we spoke about how the molecular tweezer inhibits the alpha-synuclein aggregation, but also that it restores something called the UPS, which is the ubiquitin proteasome system. And that is a system that's inhibited in the non-treatment group. So that would be the, I guess, speaking in broader terms, the people who would be affected by Parkinson's. But by restoring the system, the tweezer is able to lead to the clearance of the degenerative alpha-synuclein proteins. And then something else we said was that the study was unique in the sense that it had an in vivo procedure with the zebrafish that is reflective of what would happen in a human organism, whereas previous research was focused on in vitro models. And we talked about how it was interesting that they used zebrafish instead of mice, but we found out that zebrafish are cheaper to use in a lab than mice. So something to know. And just overall, the molecular tweezer proves to be a promising therapeutic agent for treating Parkinson's or other diseases.
Awesome. Thank you so much, Ellen. And yes, zebrafish are cheaper. So if you're looking to do research and start a lab and you're worried about money, that's one thing to think about. Anyone else from the other discussion group want to add? Maybe any limitations that you saw or any questions that you had? Okay, I think it looks like Ellen was able to get everything from both groups basically. So yeah, I just wanted to have you guys read this paper, go over it, because we are going to be discussing, or Dr. Baton might mention it a little bit next week and just to give you an idea of what he does in his lab. So if you did find any of this super interesting, I recommend you email him, ask about his lab next week. He does email back really quickly. So that's something that's cool about him that most other PS don't do. But yeah, thank you all for participating. We will see you next week. Come with questions, and it'll just be a really great way to learn about research that's being done at UCLA.
Awesome. Thank you, Archie. And thanks, everyone. See you next week. Our guest speaker.
Thanks, guys. Good night.
Bye, everyone. Take
Bye.
care.